Good day, everyone. So, our topic will be quaternary protein structures presented by Group 2. The members are ANCHETA Patricia, BALA Izaach Clark, BARINQUE Monique, and BATOCAPALA Shazmine. So, by the end of the lecture, the learners are expected to accurately define the quaternary structure of proteins, identify its structure and composition, explain its classifications, and recognize common proteins at the quaternary level and their function. Now let's have a small recap from primary structure to quaternary structure. The primary structure is the linear sequence of amino acids in a polypeptide chain. Its bond type is peptide bonds. Its importance is determine all higher levels of structure. Now the secondary structure is a local folding patterns within the polypeptide chain. Its common type is alpha helicase and beta sheets. Its bond type is hydrogen bond between backbone atoms. Now, the tertiary structure is the overall 3D shape of a single polypeptide chain. It is stabilized by hydrogen bonds, ionic interactions, disulfide bridges, hydrophobic interactions, and van der Waals forces. It involves interactions between R groups. Now, the quaternary structure is a structure formed by the interaction of multiple polypeptide chains, stabilized by the same interactions as the tertiary structure. Its example is a hemoglobin with 4 subunits. The quaternary structure of a protein is the association of several protein chains or subunits into a closely packed arrangement, and each of the subunits has its own primary, secondary, and tertiary structure. The subunits are held together by hydrogen bonds and van der Waals forces between non-polar side chains. The subunits in a quaternary structure must be specifically arranged for the entire protein to function properly. Any alteration in the structure of the subunits or how they are associated causes the marked changes in biological activity. In contrast, the first three levels of protein structures, not all proteins will have a quaternary structure since some proteins function as a single unit. Protein quaternary structures can also refer to biomolecular complexes of protein with nucleic acids and other cofactors. Enzymes composed of subunits with diverse function are sometimes called holoenzymes, meaning they are complete functional enzymes that ready to perform their specific biochemical reactions, in which some parts may be known as regulator subunits and the functional core is known as the catalytic subunit. Other assemblies referred to instead as multi-protein complexes also processes quaternary structures. A protein complex or multi-protein complex is a group of two or more associated polypeptide chains, and they are a form of quaternary structures and they are linked by non-covalent protein-protein interactions. These complexes are cornerstone or of many. The cell is seen to be composed of modular supramolecular complexes, each of which perform an independent, discrete biological function. Examples include nucleosomes and microtubules. Moreover, changes in quaternary structure can occur through conformational changes within individual subunits or through reorientation of the subunits relative to each other, in which its shape can change in response to changes in its environment or other factors. Each possible shape is called conformation, and transformation between them is called conformational change. A molecular, a macromolecule is usually flexible and dynamic. It is through such changes which underline cooperative and allostery in multimeric enzymes that many protein undergo regulation and perform their physiological function. Examples of which quaternary structure include hemoglobin, DNA polymerase, ribosomes, antibodies, and ion channels. We've talked about what quaternary structure is. Now let's look at the actual pieces that make up these complex proteins. The main pieces are called subunits. You can think of them as like individual Lego bricks. Each brick is complete on its own. It's a fully folded protein, but it's designed to connect with others. So when we say a protein has a quaternary structure, it means it's made of two or more of these individual protein chains or subunits. Now, how do these individual subunits actually stick together to form one big stable protein? They use different kinds of glue, mostly weak connections, but they add up to be a very strong connection. These most important glue is called hydrophobic interactions. Imagine oil and water. They don't mix. In a similar way, parts of the subunits that don't like water will stick together in the middle of the protein complex, away from the water around them. This helps pull the subunits together. They also use hydrogen bonds, which are like small magnets between specific parts of the subunits, helping them line up perfectly. And sometimes there are ionic bonds, which are like static electricity, positive parts attracting negative parts, also helping them hold on tight. Lastly, some of the stronger permanent staples called a disulfide bonds can form between subunits. This is a specific chemical link that locks them together in very securely, but there are less common than other types of glue. Now we've arrived in a checkpoint. So the first question is, which best describes a protein quaternary structure? 5, 4, 3, 2, 1. Second question, which of the following proteins has quaternary structure? 5, 4, 3, 2, 1. Lastly, a true or false. In quaternary structures, it refers to how multiple protein subunits and bind together. 5, 4, 3, 2, and 1. Here are the answers. For number 1, it's letter B. Number 2, it's letter C. And number 3, it's true. Now, the classification of number of subunits. Now, 2 subunits is called a dimer, 3 subunits is called a trimer, and 4 subunits is called a tetramer. A dimer is a protein composed of 2 polypeptide chains, trimer a protein composed of 3 polypeptide chains, and a tetramer is a protein composed of 4 polypeptide chains. A pentrimer is hexamers, etc. is a protein composed with 5, 6 or more subunits respectively. Now for the importance of these subunits, the first one is that it helps explain how the protein works. Each subunit may have a specific role, and how they interact affects the protein's overall function. Like hemoglobin, where subunits work together to carry oxygen. Second is that it shows how the protein changes shape or behavior. Interactions between subunits can allow the protein to respond to changes in the environment, like pH or oxygen levels. The third is that it helps understand diseases or mutations. If one subunit is damaged or missing, the whole protein might not function properly. This is key in studying genetic disorders. And lastly, it aids drug design and medical research. Some drugs are made to target specific subunits, so knowing the structure helps in creating effective treatments. Now, let's discuss the quaternary structures of proteins in terms of chemical composition classification: heterogeneous versus homogeneous. That distinction depends on types of subunits involved. Now, the homogeneous, which is a homomeric protein, all of its subunits are identical in amino acid sequences and structure. Its example is lactate dehydrogenase in some of the organisms. Now, for heterogeneous, which is a heteromic protein, its subunits are different in sequence or structure. Now, an example is hemoglobin, 2 alpha plus 2 beta chains. Let's talk about some examples of important quaternary structure proteins and their clinical significance. Insulin. Insulin is a polypeptide hormone that contains polypeptide A and B, an A chain with 21 amino acids and B chain with 30 amino acids. It is produced by the pancreas, specifically the islets of the Langerhans in the pancreas. It plays a vital role in regulating blood glucose in the body. It increases glucose storage to decrease the glucose production by the liver. Next, we have thrombin, which is also known as the coagulation factor 2. It's made of serine protease and regulates the homeostasis and maintains blood coagulation. This protein substance is produced through a conversion reaction in which prothrombin of bovine origin is activated by tissue. Hemoglobin is a tetramer heterogeneous quaternary protein. It is made of four polypeptide chains, each with its hemigroup, specifically two alpha and two beta chains. Each hemigroup contains an iron ion, which crucial in binding with oxygen. Hemoglobin is developed in cells in the bone marrow and forms an unstable reversible bond with oxygen. Arginine decarboxylase is a pentamer protein, however, it exists more commonly as an enzyme. It is an enzyme found in bacteria and plants used to catalyze the carboxylation of arginine and the conversion of arginine to agmatine. Producing carbon dioxide is also allows bacteria to survive in acidic environments like glucose 6 phosphate is a hexamer protein and is a crucial molecule involved in glycolysis. It is the first intermediate formed when glucose enters the cell and phosphorylated. It serves as a substrate for glucose 6 phosphatase in the liver, which releases the glucose into the bloodstream. Lipoic reductase transacetylase is composed of 60 protein subunits and is the key component of pyruvate dehydrogenase complex. It's an enzyme responsible for forming acetyl-CoA, which is used later process such as the Krebs cycle. In other words, it is essential for cellular energy production. We have poliomyelitis virus. It is a picoranavirus consisting of a single strand of RNA genome of four protein known as VP1, VP2, VP3, and VP4. However, these proteins assemble into a protein shell called the capsid, which includes the virus itself, and it is what allows it to infect other cells and replicate it, causing harmful interactions. Now, for our conclusion, quaternary structure is the highest level of protein structure formed when two or more folded up polypeptide chains called subunits join together to make a functional protein. These subunits can be identical or different and are held together mainly by non-covalent interactions like hydrogen bonds, ionic bonds, hydrophobic interactions, and sometimes disulfide bonds. This structure allows protein to perform complex biological functions such as oxygen transportation and hemoglobin.